Good afternoon, dear colleagues. We start the symposium, New Biomodels for Translation Medicine. Allow me to introduce Mikhail Mikhailovich Kolokudze, chair of our symposium. Dear colleagues, so we'll start this session, and it is with great pleasure that I give the floor for the first presentation to Vyacheslav Dyachuk, who is the head of a laboratory of neurogenesis at the research center of the global level at Almazov Center. And Vyacheslav will tell us about the new model systems for neurobiology and medicine. Please, Vyacheslav. Good afternoon, dear colleagues. My paper will be dealing with the new models of the systems for neurobiology and medicine. I will tell about the model systems that are currently used and what model systems we used in our research at our lab. I will dwell also briefly on such model systems as mice and fish, and on the model systems that are used in neurobiology and in medicine. Speaking about model objects, we shouldn't forget that that is a broad range of animals, plants, protozoans, viruses, and bacteria. This slide shows a lot of model systems that are used in different fields of biology, medicine, including in basic and applied studies, for instance, viruses, Lambda, bacteriophage, virus of tobacco mosaic, genetic. These are model objects used in genetic. Bacterial model object, Escherichia coli, Bacillus, Mycoplasma, the model objects for genetics. And in the last years, actively used were also protozoan, for instance, Chlamydomonas, Malaria plasmodium, trypanosomes, and fungi, Aspergillus, and yeast. Very interesting model objects are invertebrates. You probably know Caranapides elegans, Philomena lagaster. This is a nematode, and Drosophila. That is also a model system for genetics, for biology of evolution. Lately, quite actively, in neurobiology, mollusks were also used, and marine hair, that is a mollusk, also a model object for neurobiological studies. They even print were the models. Mainly, these are model systems in neurobiology. When you have a simple nervous system, it is easier to understand and to study it in the central nervous systems of the vertebrates and humans. Therefore, the scientists are using model objects, simple model objects, to understand how neurons appear and differentiate how they contact with each other in these model systems. Undoubtedly, the most popular model objects are vertebrates, particularly the group of vertebrates, Nogus. They are actively used for studying the bone marrow. As to the evolution, Dania zebrafish. Of course, that model system is used for multidisciplinary research, that is biology, evolution, neurobiology, medicine lately. Xenopus levis, mainly used for revolution biology. Gallus gallus domesticus, also a model object, that is a hen. Cats and dogs are classical objects for neurophysiology and physiology, mice and rats, multi-studies and objects for the studies of neurobiological and physiological trends. Hamsters, different chimpanzees in medicine and microbiology, and Homo sapiens. These are the objects for clinical biology, physiology, neurobiology. I would like to tell you that there are model objects, organs and tissues. For instance, such an object as ganglions of langoust. They are used as for determining the activity in neurons and extremities of amphibians. That is a classical tissue for studying regeneration, and undoubtedly we should also mention cell lines, for instance, cell lines HeLa, uterus cancer, and ST72 malignant glioma. Some statistics. These are the data of European Union for 2017, which were published in 2019. According to the data of European Union in 2017, in EU they used 9.39 million animals in experiments. For instance, 31% were mice, 15% were fish, and less than 12% were rats. And the rest were other animals shown in this slide. And the second diagram are all the animals used for the research. Most of the studies were carried out as basic studies. A great number of animals were also used in applied research. And they also studied for repertory use. Why rodents became the main object of biomedical studies? For two reasons. The possibility of genetic modification. In this slide, you can show two schemes of transgenesis. First, the first principle for which Nobel Prize was given, making specific changes to genome of mice using embryonal stem cells. When you take a plasmid or a vector from a mouse with the needed gene, and you make a preparation in the embryonal stem cell, then you isolate only those cells that were subject to transformation, and you make a microinjection and implantation into the mice, and you guess chimeral mice. That chimeral mice should be breeded and multiply to get a homo heterozygote. And in 27, for that mechanism, Nobel Prize was awarded. And the second very popular means of getting transgene animals is Cripps-Cas-Line. Last year, for that method, or that is called genetic scissors, or also Nobel Prize in Chemistry was awarded. The essence is implementation of synthesized RNA with Cas-Line protein, which inside delivers is delivered to the mice by electroporation. The first generation of mice is transgenic. It has an insert or modified gene and is a transgenic mouse. All of us know that there are knockouts, but knockouts often use transgene mice, which don't have any specific gene. But lately, new interesting model systems appeared, the so-called conditional knockouts. What does it mean? This is a possibility for the researcher to activate that knockout and to cause a certain agent within a certain time period. The sense of the genetic sense of that knockout is that you have the gene of interest that is flanked by two sides, and you have in transgene mouse CRE sequence that is encoded as a recommendation. That CRE is seen in the size FLOXP as your gene of interest. Thus, you can cut out the gene and you get a knockout. That's a very simple method of getting transgene knockout mice. And the most actively used modification of knockouts is the use of induction knockouts when you add, for instance, tamoxifen and you can activate a knockout in embryonal evolution in the early or late postnatal development in an adult animal. You add the inductor that activates the cutting out of the gene, and you get a knockout that is induction, that is called inducible knockout or conditional knockout. This is a wide type of mind with a gene of interest. If you use a conditional knockout, you can take the gene of interest, a certain promoter, tissue promoter. If you took SOX10, for instance, transcription center that is expressed in glial cells, you can kill, using that procedure, all the glial cells by killing that factor. Thus, you get a knockout which you induced within a certain time limit.
Собственно,
Well, this particular technique, the Cre-Lox recombination, is frequently used in biology. This technology is called Cre-Lox technology, which means that there is a Cre mouse, a Cre gene carrier, and this is a recombination to have the target gene manufactured. This Cre mouse is combined with the LOXP mouse, and the LOXP mouse possesses two types of cells. Those with active recombinase, and you can see there are GFP cells, and cells without an active Cre, and there's the wild type. On this slide, you can see how Cre recombinase cuts LOXP spaces, LOXP sites out, and thus a mutant mouse or a mutant mice is harvested. Genetic tracing. A case in point. To the left, you may appreciate genetic tracing. Embryos with BFBP, genetic structure activated. BFABP are glial cells. And you can see those are sutured with the tracer E10. And the second model is SOX10. SOX10 is expressed by glial cells. And if this genetic structure is activated, we can label the SOX10 cells red in red marker to demonstrate that your manipulations consider specific type of cells. Danio fish, or the so-called zebrafish, is another biological model which is very simple to operate and is quite practical in developmental biology because the embryos are transparent, they develop frequently, very fastly, and there is fast reproduction, and this biological model is not costly. So Danio revio fish is frequently used in biomedical research. It's a very convenient object because you can stain the embryo or the egg or the fish with different antibodies and do the fish analysis. The most fascinating results can be obtained when you operate a transgenic animal and then stain it with different types of antigens to be identified. A most fascinating method of genetic tracing is associated with tamoxifen. You add tamoxifen, and in 44 days, you look at the fish to find out what happened with the stimulated cells. Here you can see the SOX10 promoter expressed by glial cells. If you add tamoxifen, you activate the tracer in these particular cells. And these animals actually allow to stain a particular type of cells. But then you may add extra staining for other markers. And this is highly useful to investigate stem cells and their potential. So fish are a great biological model for pharmacologists. You can add the activator into water in vitro and have a look at what's going on to the fish. Here you can see what happened to the zebra bow zebrafish, and you can see the sympathetic ganglia. And when the glial cell inhibitor is added, we inhibit this receptor in glial cells, and we see that the sympathetic ganglia and the DRG cells are either depressed or inactivated, or they are absent at all. So these cells can be differentiated into sympathetic and sensor neurons. As for zebrafish, I cannot but mention the CRISPR-Cas9 technologies used in 90% of laboratories that deal with zebrafish. This slide is borrowed from a paper, and you can see the neurons and the chromaffin cells. And if we inhibit the OLIG2 transcription factor, which is expressed for motor neurons, if we kill this factor by CRISPR-Cas9, the differentiation of neurons, both sympathetic and DRG neurons, is disrupted. And this is identical to adrenals in mammals. And we see that the number of such cells is decreased significantly. Such tools allow neurobiologists and doctors investigate specific genes and gene ensembles or gene orchestras responsible for the development of neural system and other tissues and organs. This is it. Thank you for attention. Thank you, Vyacheslav. Questions? We can have one short question according to our time frame. Well, you mentioned that the destruction of glia disrupts sympathetic and sensitive ganglia roots. Are there other approaches that allow for better differentiation and can differentiate between sympathetic ganglia and DRG sensor neurons? Are there approaches that can block only parasympathetic ganglia? Thank you very much for the question. The ganglia cells in zebrafish can migrate from the spinal cord to the trunk. And when they pass through DRG, they first express DRG neurons, and then they also express sympathetic neurons. But timing is all the essence here. This process is an early developmental process, and our screening data show that the glia migrates from the cord and forms DRG neurons, whereas some part of glia produces sympathetic neurons, but the time difference is 4 to 5 hours. First DRG neurons and then sympathetic neurons. We can't split these two processes apart, but we need an extra tool to stain specific glial subpopulation. So far, this is technically impossible. But in the future, I believe that here we are confronted with different types of glia that produce different types of neurons. Thank you very much. Really exciting. Dear colleagues, I would like now to move on according to the agenda and give the floor to Marina Vasutina. Daniel Rario is a model for studying the genetic mechanism of dyslipidemia and atherosclerosis. Please, Madam Vasutina, the floor is yours. Thank you. Distinguished colleagues, good afternoon. As a follow-up to my previous presentation that was devoted to biomodeling in atherosclerosis, I would like now to present my talk on Daniel Rario as a biological genetic model for dyslipidemia and atherosclerosis. I'm going to repeat some slides. Here you can see the overview of different biological models for atherosclerosis over the last 100 years, starting from 1925 with the step of 5 years. So you can see that overall, PubMed has less than 100 papers that can be sampled out for Daniel Rario and atherosclerosis input. So in Science Cell, we see that in late 2000, we can see that genetic models started to dominate regular standard environmental models. Daniel Rario, a zebrafish, is a valuable model to study different biomedical processes because this fish has transparent embryos, transparent eggs, which allow us to study the egg development and to do staining. You can see the number of Medline papers. Most of those belong to the US and Europe. Genetic modifications of Danio fish go back to the early 2000s. It is in the early 2000s that the number of papers using this biological model started to increase using RPC2 mutation and LDLR mutation. The standard approach is to study LDLR mutation in rat and in rodents. However, Daniel Rario allows us to investigate visceral obesity, fatty liver, retinopathy, cardiomyopathy, and brain disorders, as well as hyperglycemia-induced wound healing defects. Daniel Rario have a lot of similarities with vertebra and can serve as a model for different human diseases. The advantage of Daniel Rario is that it is a unique biological model, a vertebra that allows us to perform broad-scale chemical and biochemical screening. One pair of fish can produce up to 200 to 500 eggs with homogeneous background. And this allows us to have a 100% homogeneous sampling. Phenotypical screening ensures high transparency of eggs. Therefore, we can easily integrate fluorescent staining into these eggs. High-resolution investigation of specific bodies and similarities in terms of biological and physiological parameters make this biological model indeed invaluable. This allows us to perform in vitro studies on larger animals. This model is highly accessible and allow for different types of genomic, molecular, and visual screening. As a test system for atherosclerosis modeling, we could focus first on organizational issues and then on scientific issues. As for organizational issues, well, some lines show low productivity without any 100% survival of the offspring. This is for rodents. As for Daniel Rario, the eggs are transparent, easy to keep in automated systems with specific water preparation and water recirculation. Well, actually, the complexity is similar because except for the fact that water is more prone to different pathogens than the environment of rodents. Mice are resistant to exosomal atherosclerosis modeling factor and may not respond to diet, speaking about genetically modified animals. So to produce a markedly significant atherosclerosis in mice, specific genetic modifications are required. To compare, Daniel Rario produced very good results without any tricky genetic modeling. Just a diet would be enough. Daniel Rario is indeed an ideal biological model to modify or to produce a biological model of atherosclerosis. As for similarity between Daniel Rario and humans, well, mice do not produce an unstable atherosclerotic plaque. Quite the contrary, mice produce a stable atherosclerotic plaque. However, a plaque rupture is very exciting to study its pathogenesis. On top of that, mice rarely demonstrate any disorders of coronary arteries. On top of that, mice do not allow us to extrapolate the thermal regulation results onto the human model. On top of that, mice have a different reproductive system. Daniel Rario, on the contrary, are very similar to humans because they can produce an unstable atherosclerotic plaque which is induced by low-density lipoproteins. Considering that mice have different lipoproteins, they are resistant to particular impacts that humans are prone to. In default, fish are more prone to hypercholesterolemia than mammals, and this makes them also prone due to specific carbohydrates metabolism. Therefore, fish are a much better model than mice that do not express fatty tissue receptors. Genetic manipulations in case of Daniel Rario are simpler because Daniel Rario are specific in terms of reproduction and genesis. As for the methods, we should focus on the fact that Daniel Rario eggs are transparent and adult animals can also be transparent, and therefore we can monitor changes and disorders in organs and tissues.
If you want to look at the inner organs in mice, we have to use CT MRI system or remove the mice from the experiment, seeing what is inside. This is also an advantage of fish over mice. However, it is rather difficult to take blood samples from a fish if we want to have the lipid profile, geochemistry, which we can get from biological liquids. For mice, it is easy. Now we speak about the genetic models of fish which are used in the sphere of atherosclerosis. I would like to start with Kantar's the investigation, that is dose-dependent effect on inflammatory biomarkers and lipids. It is interesting that groups of patients received placebo or a drug, canakinumab, that is human monoclonal antibody targeted at beta-interleukin-1, and that drug doesn't affect concentration of proteins of low density or high density proteins, triglycerides, but it affects C-reactive protein and its content, which reduces significantly. It is interesting that further, when we track further cardiovascular risks and events, neuroatherosclerotic cardiovascular events, we can see that people who receive the drug less frequently have such consequences. The risk of complications of cardiovascular system is reduced. Thus, it shows that even low level of LDL does not always guarantee the reduction of risks in terms of cardiovascular system, because autoinflammatory disorders are a risk factor for development of atherosclerotic alterations and cardiovascular complications that can be important even in case of a low level of protein in the blood. Our Italian colleagues described one of autoimmune diseases, psoriasis. It is characterized by penetration of the artery, stenosis, and formation of the plaques with normal or low level of PNT. So now, in the sphere of atherosclerosis, they also take it into account. And for the study of these mechanisms, we can use genetically modified fish, chronic inflammation through activation of NLB3 inflammasome. Beta signal route is an important factor of development atherosclerotic cardiovascular diseases, being started by accumulating of intercellular cholesterol in cells. Publication of Italian colleagues, manipulation of the gene responsible for the process on fish were carried out under the research on antibacterial immunity in vivo, and the pyrin domain of that family containing that chromosome, inflammasome, is one of the most characterized in mammals and people. In the study, there was molecular and functional identification of homologue Daniel Rario, and due to lockdown homology and generation of knockout and Daniel Rario SC, we could characterize the function of that homologue in antibacterial immunity in vivo. This is a scheme on pathogenesis of that structure, that signal pathway in referring to atherosclerosis. We said that the publications which generalize fish, zebrafish and atherosclerosis are not numerous, and the effect on atherosclerosis using zebrafish model has not been finally studied. It would be interesting to analyze the gene and the process of atherosclerosis model on Daniel Rario, APOC2 deficient. one of the vectors of further development for the model dyslipidemia on fish. However, it is classical one of first mutations in atherosclerosis obtained on fish. People deprived of OPC2 have familiar chylomicronemia characterized by increased level of triglycerides and the recurring episodes of pancreatitis. Mutant denuria demonstrate features of deficit of OPC2 of man, low activity of lipids in plasma, and severe hypertriglyceridemia. Visualization of vascular network results in accumulation of lipids and macrophages, which is a feature of formation of atherosclerotic plaques. And we can easily visualize a vascular network in fish using fluorescent markers. We can analyze this transparent larva of fish. That is a convenient model that makes it possible to assess the process in vivo. That's a great benefit of the model. That model of dyslipidemia might be studied to study extravasation, oxidation, and absorption of low-density proteins by the model LDR deficit on Denurera. We have knockout mice and LDR deficient Denurera. Zebrafish mutant in LDRL are biological models for studying hypercholesterolemia and accumulation of lipids and blood vessels. In fish, it is easier to consider the early stage of development of atherosclerosis and formation of the plaque because in our probability, in order to model late stages up to destabilization of the plaque rupture, these structures have not yet been found. Probably they exist, and we have to determine the gene of interest and to expand the sphere of research. There are less than 100 publications, and there are a lot of questions not answers. This model is characterized by development of moderate hypercholesterolemia at normal nutrition. However, short-term 5-day nutrition with high content cholesterol results in hypercholesterolemia and accumulation of lipids in vessels. That is an important model. Besides, there is a list of genes of interest. It is not complete. Now, other genes were studied that might be interested from that viewpoint. That table shows the genes and the mutations which are studied for different animals, but we focus on mice and fish. In fact, they are much more numerous. The brightest mutations, and the main accent is on decreasing or increasing the level of certain substances. For instance, triglycerides. For increasing triglycerides, there is a great number of models. They are presented here. You can create a lot of models, and we don't know how many genes of interest we might associate with zebrafish, for which a genome is segregated. There is a broad range within which we can work and develop. So all the researchers I ask, and the doctors, I ask you to take into account that we have a wonderful tool of model animals. Some models are done quickly and understandably and can demonstrate a lot. Data are extrapolated, but the fact is quite interesting, and that can be really helpful in further development of clinical aspect in treatment of atherosclerosis. Thank you for your attention. Are there any questions? Thank you, Marina Lvovna. Unfortunately, we don't have much time for the questions, so we have passed on immediately to the next presentation. It will be presented by Lada Alexandra Murashova. She will speak about the place of organoid models in basic research. Organoid models, a new tool for basic research. Good afternoon, dear colleagues. I would like to present my brief presentation about organoid models. That theme has been developed in the last 30 years quite actively. And the further we go, the more complicated the models become. And the further we go, the more we can do within these models. The currently available cellular models, organoid is a 3D model, which is outside the body of the animal. Now we have classical 2D cell cultures. They are easily available, they are rather cheap, and we can carry out investigations with a high speed and low toxicity. The disadvantage of 2D classical models, the cells, while being attached, change their transcriptome profile and cannot behave as in the organism. Besides, 2D cellular structure doesn't make it possible for a long time to cultivate cells of bone marrow, immune competent cells. The so-called 2.5D culture cells, that is a plastic on which there is a sponge, and they put cells on the sponge, most sensitive cells, and so to cells, nanny cells, which accompany them in tissues, and 3D cultures. 3D cultures are self-organizing spheroids. This is a population of the cell culture with one type of cells placed in the Henning drop, or under conditions when they cannot attach to the plastic adequately, and thus they form spheric structures. In the slide, that is the third image from the up. They are quite convenient, and we can assess the behavior of the cells in the 3D structure. Besides, they are less or more or less optical transparent, and there is no intergrowth of spheroid due to the limited contact with the medium, with the nutrients, and limited diffusion within the spheroid. Necrosis appear. There are serious, that is a serious limitation in the work. Spheroids on scaffold, they are formed or printed. They are manufactured on an industrial extent, nets and sponges to which cell cultures grow, and 3D structures formed in organoid models. These are classical organoids of normal tissues, embryoids. These are the structures comprising embryonal cells that can differentiate, but they do not develop to the full organism. Tumoroids, 3D organoid structures, which have all properties of tumors from which they were obtained, and a 3D printing, but that is with a scaffold and 3D structure or organoid structure. Organoid per se can be characterized as a self-organizing, self-renewing 3D cell culture distinguished from the primary tissue or induced pluripotent stem cells, have functional features of the tissues from which they were isolated with similar transcriptomes because the cells neighbor each other and they can, so to say, communicate and isolate to the environment growth factors, cytokines, and mediators which can excrete in a living organism. The scheme of obtaining an organoid, the simplest scheme of organoid from a small intestine, we remove a part of an intestine from the animals. We fragment it mechanically, then we use the enzymes, and then we form three possible versions of further development of the organoid. We take either cells and place them into the medium, and they self-organize into these intestine fibers or crypts, villi or strips. We can isolate stem cells and induce them towards growth, and with the time they'll form a normal organoid. We can also take pluripotent stem cells from amniotic fluid. We can take, if we're speaking about animals and organoids, we take embryos, or at an early stage of development, we can mince them into cells and grow by induction with different growth factors. The organoid which we require in our study. Currently, there are a lot of organoids practically for all organs and systems of the organism. Organoids of the lung were used right now during the pandemic for studying interaction of coronavirus COVID-19 with lung tissue cells. The same organoid of the intestines was used in the study to determine the interaction of the virus with that organ. Organoid can be also produced for the intestines from the small and large intestines, depending on the part you need. Organoids of liver, biliary tracts are also quite good in the study. Pancreal organoids, organ glands, and the secreting enzymes of the inner brain. Such organoids of the brain have already been obtained. They generate alpha rhythms. Prostate, kidney, cardioid. Cardioid is an organoid of the cardiac muscle. There are cardioids that can contract spontaneously and independently, and each cell has its own rhythm. Tissues of the women's reproductive system, endometrium, tube, and tumoroids of some solid tumors, for instance, colorectal cancer can be easily studied. Advantages of organoid models versus 2D and 3D spheroids. They compose cells of different differons, so this is not one population of cells. They model interaction between the cells in the organ source of the tissue. If we take the intestines, we can model the interaction even with microflora of the intestines. If that is a microfluid system, on the one hand, we have microfluid system directed to crypts or intestine villi. On the other side, it is directed. There is an own plate, and we put those crypts or microflora, and we see what would happen, how it interacts with normal pathological microflora. They will excrete mucin, they will die, as in the organism, and they will synthesize interleukins, asking the immune system for help, so which the organized system doesn't have, but nevertheless, the behavior would not change. It is rather simple to edit the genome, particularly with pluripotent stem cells. We can sew into organoid models fluorescent markers, and we can observe in dynamics how the cell and its clones develop further. We can use in organoids species-specific biochemistry and physiology of the tissue. For instance, in the liver tissue, in human liver system, for instance, there, in case of decontamination of the toxic substance related with glucuron acid and sometimes sulfonation, sulfonating metabolism would be preferred in pharmacological substances. In the live organism, we have to take it into account. Organoid model makes it possible to use the tissue of the species that we're going to use in future work, a rather low cost versus animal models, including the genome editing, and the waste of organoids is less. We process control that process to a greater extent than for cats, good dogs, pigs, mice, and fish. Another possibility is bring together different organoids into one microfluid system to model mini organism body on a chip. It is not quite full-fledged, but due to microfluidics, we can signed a signal of the set of organoids via liquid systems, liquid channels to the other system. And that system exists, so we can connect about 10 organs. It's called body on a chip. And we can also connect into this system tumoroids and analyze how the neoplasms, for instance, ovarian cancer, would interact with pulmonary tissue biochemically. Restrictions of the models. There are certain restrictions of the model, of course. First, the lack of natural microenvironment. Even if we are speaking about pancreas, liver, or intestines, when they are inside the body, first, the organs have certain pressure, which they exert on each other in the cavity. Then there are some stromal features, and the connective tissue might have fibers that also affects the communication with fibroblasts. That is very important for the organ stromal fibroblasts. It is impossible to model the immunological response to the full immunological response. Intestinal organoid might use macrophages, neutrophils for health, but no one would help him and they will die alone. Lack of nervous regulation, they don't get a normal signal, and there is no feedback with the brain as it would be in the living organism. Lack of complex endocrine regulation. We can affect specifically using a hormone, see how the cells and tissue will behave, but we cannot create that feedback, that fine balance that of a living body. As to the organs and tissues, human organs and tissue, these are ethical and legal aspects which our organoid models are not sufficiently developed.
So what are the aspects for organoids to be used? Well, we can monitor the development of an embryo, or we can look at the cell migration. We can add a retinoid acid or remove it, and look how this impacts the migration. We can do these manipulations with cells of large mammals. On top of that, we may add tumoroid modified cells to healthy organoids and look at their interaction and look at the changes in the signaling and cellular morphology. Well, this is in vitro, and it will be different from in vivo, but this allows us to have more information than in 2D space. So I think that to reduce this to the minimum, the organoid microfluid systems have the future. Here you can see a cardioid, a self-organized and self-contracting cell that was grown in a cytokine-rich environment. This article was published in 2021, and it's an exciting one. I really recommend to read it. Thank you so much.
I would like to present and introduce Professor of Dental Medicine Kai Fried from Karolinska Medical University. Kai is top specialist in neurobiology and dental medicine. Kai, are you hear me?
Yeah, I can hear you.
Fantastic. Let's start, please.
Okay, so I just put on my presentation. is this presentation visible?
Yep.
Can you see my screen?
I see you, but not presentation yet.
So how do I do in order to, because I'm not host? I have to be able to show my slides.
You have to share screen, as I remember.
Yeah, but I can't do that.
I will ask the technical specialist. One moment, please.
And thank you very much for inviting me. I'm Kai Fried from Stockholm, Sweden, and I would like to discuss with you just for a short while something about mesenchymal stem cells and a model for biomedical research, which nature has given us in the form of a continuously growing tooth, where we have mesenchymal stem cells that are continuously replenished and makes it possible for an organ, in this case the incisor in the rodent, to grow throughout life. And this is a schematic picture of a mouse lower jaw or mandible, where we can see the incisor, and this is what we see in the mouth. And during the lifetime, the mouse is continuously gnawing this part of the tooth so that it will be abraded, but it's continuously growing from the apical end, the other end, in order to replenish the tissue that is being lost through this continuous gnawing. And the tooth is a complicated organ. It's built through different tissues, and it's formed through a very complicated interplay between different types of initially embryonic tissue. And just to show you something about how teeth are being formed during development and how this bears to stem cells and the life cycle of stem cells, I just want to show a slide describing how teeth are being formed. And as I said, it's an interplay between tissues, in this case between the oral epithelium in the fetus, in the embryo, which is epithelial, and the connective tissue beneath, which is the mesenchyme. And this mesenchyme is derived from the neural crest, so it's related to the nervous system. And then epithelial part where the tooth is going to be formed, it starts to grow downwards and forms an invagination. And in different stages, then the tooth is being formed from these two initial tissues, the epithelium and the underlying mesenchyme, which is from the neural crest. And here we can see thickenings, and we can see vaginations of the mesenchyme. And this part, the epithelial part, will form the enamel, the hard tissue that's on top of the tooth. And the mesenchymal tissue that's derived from the neural crest will form the inside of the tooth, which is the soft tissue, the pulp, and also the second hard tissue of the tooth crown, which is the dentine. So these are the different stages, and this is the postnatal stage afterwards. And here the tooth, and this is from a mouse actually, but it works the same way in human. And at this stage, the tooth starts to erupt. and becomes visible in the mouth. And this image also shows a very close vicinity between the nerve fibers, which are depicted here with black lines, and nerve fibers actually surround the tooth for a long time without actually entering the tooth, which only takes place at the stage when the tooth starts to erupt. And just to show you, sort of, it's not real life, but what it looks like in the microscope. So here we can see from a fetus, a human fetus, this epithelial invagination here, which eventually will form the enamel and the neural crest-derived tissue inside the tooth, which will form the dentin, the other hard tissue, and also will form the soft tissue inside, and also a follicle around the tooth. And we've been studying this process in a model organ, which is the mouse incisor, which is formed in basically the same way. So it's also an epithelial thickening here that grows downwards, and then it has slightly different shape than what I showed you in the previous slide, and it will form this elongated tooth bud or tooth anlage, which eventually will start to produce hard tissue in the form of enamel, as is shown here, and the dentine. And but the same tissues that interact in human when forming a tooth is also interacting in the mouse. And here it's just the shape that differs. In the mouse, we have 3 teeth that the mouse uses to chew, which are molars, and the incisor, it continues to grow, and we don't have an equivalent to that one. But this fact that this continues to grow throughout life makes it interesting for studies of this general process of formation of the tooth, which can be done not only during development, but also during adult life of this animal. And in this picture, this image here is more or less what you see in the next pictures, just that it's standing up in this image. And this is a slide showing the outlines of the tooth here. So this part here will form enamel and eventually also dentine. And this is the inside of the tooth. And we made previous studies a few years ago where we can see that a lot of the tissue inside the tooth, which I said is derived from the neural crest, is also derived from supporting cells from the nerves. And in this picture you can see in red nerves surrounding the tooth, and in green cells that are covering the nerves, so-called Schwann cell precursors that actually leave the nerve and contribute to form the tooth. And these cells actually leave the nerve here, and in green you can see that they inhabit the inside of the tooth, and they also form the cells that eventually will start to produce the other hard tissue, dentine. And this image, it's specific because it shows us how the nerve will actually contribute to the formation of the tooth together with neural crest cells that have migrated here. But it also shows the area here between those folds that came from the epithelium, and this is an area actually where the mesenchymal stem cells, where they have their origin or their initial site, either if they're from migrating neural crest or if they have arrived through riding along the nerves here. And we can see in this picture how these nerves eventually seem to be found in different places and move along to their final destinations. And this image, it shows schematically how the Schwann cells that will leave the nerve, how they eventually form clones here that will traverse through the nerve and occupy different sites within the tooth. And this is during development. And much or exactly the same way neural crest cells that have migrated here and not arrived through the nerve, they will also form these kinds of streams through the developing incisor. And this is development. So what does it look like during adult life? So then we would have to look upon this schematic image again, and we have this incisor. And now, mind you, this is an adult tissue. This is the lower jaw of an adult mouse. And the area we're interested in is the area where new tissues are being replenished, are being formed. And this is the so-called apical area, and it's framed here. So you can see it within this square. And when we look in the microscope, we can see this area here between those sort of folds here from the epithelium, which is this one, CL2 and CL1 in this picture. And in green, we can see these Schwann cells, these very primitive neural crest cells that arrive on the nerves, how they gather in this area, which is actually the stem cell area here. And we can see some of them also along nerves here, but when this one is magnified, you can see that there are lots of these very early mesenchymal stem cells within this area, and they are traced here through so-called genetic tracing, so with a specific Schwann cell marker. And we can see that they inhabit this area here, which corresponds to this area here. And this genetic tracing can be done, the so-called clonal analysis can be done in different colors, actually, so you can follow different clones, individual stem cells and their progeny, what comes from these individual stem cells. You can follow them and see how they actually form these streams that I talked about, and then eventually the fate of individual stem cells. And this is an example of that. So here is actually a nerve, and there are these primitive Schwann cells that have left the nerve here, and we can see the clones that belong to one individual Schwann cell here, colored in red, and it will actually eventually continue and form cells that will produce hard tissue. So these are mesenchymal cells that will differentiate into cells that will form hard tissue. And here's another clone which is colored in yellow, and we can see the relationships between these different clones. So far we can see that in this area, in the growing tooth, there are stem cells that are derived either from the nerve, well, in the adult, mostly from the nerve, or from remaining more or less silent or quiescent stem cells within this area. So the next question, of course, would be, so what decides why a stem cell, here depicted, for instance, in red, green, blue, or black or grayish, what decides what the fate will be, and what different steps would they have to go through, and what will influence them in order to go in a certain direction? So we can see in red here some cells that go, and some of them will produce pulp cells within the tooth, and some of them will actually be these dentin-forming odontoblasts along the wall here. And it seems that the vicinity, the closeness to the wall of the tooth here will actually decide whether one of these Schwann stem cell progenies will form these dentin-producing cells, or if they will become mesenchymal cells within the so-called pulp, the soft tissue here. So these ones that are red here, and these that are black or grayish here, they are close to the wall, and many of them will become these odontoblasts here, while these which are more or less in the middle here, they will eventually become only pulp cells, that is mesenchymal cells or connective tissue cells, and also those in green here. So along these axis here, it's important where the cells actually are found or placed. So, of course, a lot of questions are, of course, being raised when you look at a picture like this. So this is just a repetition of what's taking place during tooth development. But when we look at this area here, which is just between the folds of this epithelium, which will produce enamel, when we look in this area, we have a stem cell area here quite clearly. And this is supposed to depict the stem cell here. And so the question is, if the stem cell will start to divide and is close to the wall here, for instance, what factors will influence the stem cell in order to make it differentiate into whatever final fate it will acquire. And there's a lot of signaling here, of course. We have some major players here, Wnt and fibroblast growth factor, BMP, and sonic hedgehog. But we don't know much yet about exactly what's taking place in detail in order to differentiate these cells along different paths. So then you would like to know more about how they are actually the characteristics of these cells in the different stages, which is quite difficult when you look at during stem cell activation, if you want to look at transcription factors and other genetic components. And of course, the traditional way would be to just look at the bulk RNA input, and then you would have an average gene expression from all cells, but you would not know which cells that express what, and you would not know at what strength, of course. So I'm just trying to simplify very much the procedure of single-cell transcriptomics, which has been used here, where single cells can be extracted, they can be identified uniquely through a barcode, and then they can be grouped into different clusters depending on what they express. And this has been done here in this area that I have described to you, this apical area in the growing incisor and also in the developing incisor. So this shows that this area here, which is the apical continuously growing area, which replaces tissue through a lot of cellular events, this tissue will give rise to mesenchymal cells that eventually will be differentiated into pulp cells and also to odontoblasts. A similar thing happens with the epithelium in order to produce enamel, but that's not the topic here, so I will not talk about that. But if you do these single-cell transcriptomics experimental procedures in this area, you will be able to group through bioinformatics eventually, to group all the cells here into clusters. And you will have a plot like this, a Tisnig plot, where we will find the mesenchymal cells here in this area here. And then there are lots of other groups of cells, which I will not talk about here. For instance, immune cells and endothelial cells and glial cells along nerves and perivascular cells and other cells. But we will only focus on what's present here in the mesenchyme and what will happen with those cells later on. So we are in this area, and we would like to know more in detail what's taking place here and along what paths these different cells differentiate. I'm sorry, because now I lost this one. I will go back. And by grouping the cells like this into clusters, it's also possible to see that there are different types of cells within this apical area, although you're still just focusing on this mesenchymal cell area. And by finding what these different groups of cells actually express, it would be possible to pinpoint the different groups of cells and localize them. And if you look, I'll go back to this one. If you look in this area, you will find one group of cells that actually are more or less a constitutive type of cells, which are more stromal in their nature, and then they're probably to support the stem cells, and you can find one type which has renewable properties like Thy1 and Gli1, which is restricted to this area. And then there's another which is has typical markers of dividing cells. For instance, FOXD1 expression and FGF3 expression, which you will find in this region. And then a third one, which actually has started to differentiate. So you would have one type, which is more or less a kind of a stromal cells only, one type with a quiescent stem cell, one type which is dividing, and one type which is already starting to differentiate. And by knowing the markers of these cells, you can also identify them by staining with different RNA markers or protein markers. And if you look just as an example at the FOXD1 expressing cells, which are the dividing cells, you will find them here in marked with tomato red through lineage tracing, genetic tracing. And this is the epithelial fold. This is the inside of the tooth. And we can see that these cells are being found here along these epithelial folds and a little bit further on, and then later on also in odontoblasts. And this is a magnification of the area here. And by continuing along those lines, then it will be possible in this area, which is the apical pulp area here, it would be possible also through what is called the RNA velocity method, which is a way of measuring strength of expression of different genes over time. You can also predict what will become of different types of cells. So what we can see here is the apical pulp, and then how it will give rise to odontoblasts and also to distal pulp cells. So I will not go into detail, but it seems that they will acquire a final fate, and that there are transient stages, and they will eventually acquire a program that will differentiate them to a final fate. And this gives enormous amount of information about transient states and what they express at different stages along these paths, which I will not go into detail here. It's just a way of expressing new ways of actually understanding stem cell behavior, and then what triggers division and what eventually is the path towards differentiation. And this is schematically shown in this picture when it comes to the mesenchymal part, and this one is the epithelial part. And it makes it possible also to see in detail, for instance, when it comes to the odontoblast, which produces dentin, to understand in detail the gene expression at the different stages towards the final differentiation when dentin starts to become produced. And I will not go into detail here. It's just a way of showing that we can see now every step, more or less, in terms of gene expression in between stem cell activation and the final tissue. And of course, this is, as I will conclude with, these are extremely important things when understand how to regenerate tissue. So the issue then, of course, is mouse and human. Is it the same or is it different? Because that's what was the topic of this session, the new biomodels. And if we want to have a model in an animal, we want it to be similar to what is found in human. Otherwise, it will be less useful in terms of developing new ways of regeneration or new ways of testing drugs. So the same kind of approach has been done with human teeth, and the human molar tooth is quite similar to the mouse molar, not the mouse incisor, but the mouse molar. And the molar has been studied, the adult molar and also the growing molar. And this image more or less shows that in this picture that we have in the apical part of the growing molar, which is sort of corresponds to what we can see in a mouse incisor in the apical part. It has its specific signature, which we can see more or less here in this region when these cells are being grouped in through single-cell transcriptomics, while the adult cells are in another group. And this can be seen more in detail in this parallel image where they also, it's written out exactly where they are. So these cells are from the apical papilla in a growing tooth, and this is the adult molar here. So the issue would, of course, be, are they similar, these, to what is seen in the mouse? And very generalized, these are quite similar to what you can see in the apical area of the mouse, and these are more or less rather similar to what you can see in the distal part of the mouse incisor when they have differentiated. So there are certain similarities between mouse and human. There are important similarities, but there are also important differences at the end of the day, which is expected also, of course, since mouse teeth and human teeth, they are not identical. There are specific differences, and that would be reflected also in the gene expression and how these single-cell analysis will, the outcome of these analysis. So to conclude, these studies have produced an atlas of human and mouse teeth during growth and during differentiation. And the purpose of this is to understand exactly mesenchymal stem cell behavior and differentiation during development and also in adult life in order to make use of these to understand better how we will develop new ways of regeneration of tissue and new ways of also perhaps also testing drugs in these models. And what we have seen is that the complexity of the continuously growing mouse incisor is far, far larger than what was anticipated earlier on. And this is the strength of this method, of course, because we can see what we cannot see with previous methods. We have a good model here of the cell dynamics, which actually enables unarrested growth. It's continuously growing all the time. And we now have the data which shows how this goes on. And it has pinpointed what type of cells that we find in this region where there is, in the apical region of the continuously growing incisor. We have cells that support the region, and we have progenitors, and we have differentiated cells. And we have also looked closely at the human and mouse, and we see that there are important parallel mechanisms and differentiation patterns, but there are also differences which we have to also study further. And as I said, this might lead to possibilities both to isolate the actual stem cells in rodent teeth and perhaps also in human teeth. And it might also show ways where we can find ways of activating in situ stem cells and also encourage regeneration or growth by stimulating local cells to produce new tissue that has been lost, for instance, through trauma or through disease. So So thank you for attention. I would just acknowledge, first of all, Igor Igorovich Adameiko in Vienna and in Stockholm, and then also Jan Krivanek in Brno and Peter Harchenko at Harvard and Ruslan Soldatov. So I thank you for your attention.
Thank you, Kai. It was fantastic talk. Thank you very much. Well, so the next speaker, I would like to introduce Professor Andrei Chagin from Gothenburg University and Karolinska Medical University, Sweden, Stockholm. Please, Andrei, are you hear me? Hello, how are you? Let's start.
Can you hear me?
Absolutely.
Can you see my screen? I can't hear anything.
Yeah, we see your presentation.
That's surprising, but Yes, can you hear me? Can you see my screen?
Yes.
Good. The sound was disappeared for a while. First, I'd like to thank the organizers for inviting me to give a talk. So I'll talk about models and approach in skeletal tissue regeneration. And as you can see here, there are quite a bunch, there are several stem and progenitor cells located in the skeletal tissue. Here is in the bone. You can see that the articular cartilage contains kind of progenitor cells which give rise to articular chondrocytes. There are stem cells being reported at the top of the growth plate, which give rise to all chondrocytes in the growth plate and a subsequent bone. There was also been reported cells, stem cells locating along the blood vessels, which we know as bone marrow stromal cells or mesenchymal stem cells of bone marrow, which give rise to trabecular bone. And there are also cells, stem cells being reported in the periosteum, which give rise to cortical bone. So what's uniting all these stem cells is that not only bone, but it also that these cells, or the definition that these cells are kind of stem and progenitor cells, comes from the lineage tracing. And this is what I would like to focus this presentation. And I'd like to focus it on lineage tracing because my laboratory in Moscow and the laboratory of our chair session today, Dr. Dichuk, brought about 10 different transgenic mice to Russian Federation, and I will describe some of them. So, Krylov system has been mentioned by Dr. Dichuk and likely by Dr. Kai Fried today. I'll get a little bit more details about that. So it consists of two elements. It's a LOXP site, which is very simple. There's 34 base pair of DNA sequence. And there is a Cre protein, which also comes from bacteriophage. And it's 343 amino acids, and the Cre is just a DNA recombinase. It's recognized these two LOXP sites and cut everything in between. And I will also mention about Cre-ERT. It's the same as a Cre protein, but it's fused with estrogen receptor. And I'll talk a little bit more about that. So how does it work? If we have a LOXP site and some DNA part and another LOXP site, the Cre will recognize those two LOXP sites, bring them together, and cut everything in between. The cutted fragment in mammalian cells usually disappears, and we will get the DNA sequence without would have been inserted between these two LOXP sites. In relation to CREA-LOX system, to CREA-ERT, it's slightly more complicated. So the CREA is fused with estrogen receptor ligand binding domain, but the domain is mutated in the way that it cannot bind endogenous estradiol, but it still can bind tamoxifen. So how does that work? When the cell is expressing CREA-ERT, it stays in the cytoplasm because estrogen receptor part keep it in cytoplasm without a ligand. But when we inject tamoxifen, it binds to this estrogen receptor part, and the whole protein translocates to the nucleus and can induce CREA-dependent recombination. So how would we use it for? We have a reporter gene and a ubiquitous promoter, but it contains a stop cassette in front of it, surrounded by LOXP sites. So when this cassette is removed, the reporter starts to be expressed, and the cell becomes visible. The reporter can be anything. It can be GFP, tomato, fluorescent protein, it can be non-fluorescent protein, just any protein which you will be easy detect. So you can see that particular cell where recombination happens, and we start to produce that. So practically how it works. We have a CREA transgenic line and a CREA under tissue-specific promoter or cell-specific promoter, some whatever cells you are interested in, and you know that these cells express this particular promoter, and this particular promoter is active in those cells. So then you need to have a CREA which is under that promoter, regulated, and then it's transgenic line, right? This transgenic line, and you cross it with a reporter transgenic line. And we have now several reporter lines in Moscow, and we have several reporter lines in Almazov Center, in fact. And promoter-specific CREA you can either make yourself, or usually what people do, they just look around. There are about more than 1,000 different CREA reporters lines, so there are depositories where they can be purchased for a reasonably low price, or in collaboration can be obtained from people who made them before. So we have several tissue-specific promoters now also in Almazov Center and in Sechenov University in Moscow. So when we cross this mice, we'll get a mouse where this CREA is expressed in a tissue-specific manner, and the reporter gene is expressed. Every single cell contains a reporter gene, but it's not activated because it's stop codon. So how we practically use that? We can, let me see if I can play the video. Yes, like that. So here is example that we take. We want to label the Schwann cell precursors, which been mentioned today already a couple of times. So we take PLP1 driven CREA, as you can see here, and it's CREA-RT, so it's inactive until we inject tamoxifen. And we cross those mice with reporter mice, which express the YFP. And then we inject this embryo with tamoxifen at E11.5 and collect at 12.5, 1 day later. And we can see the Schwann cell precursors as green cells located along the nerves, and the nerves are stained by immunohistochemistry with a 2H3 antibody nerve. So you can see nicely how this Schwann cell precursors are localized along the nerve fibers. How we can use it for addressing scientific question. So here's the same embryo, PLP1 CREA-RT, labeled at 11.5, collected at 12.5, but just in a section we see that the Schwann cell precursors located along the nerves, but nothing is located in the cartilage. Cartilage is stained by SOX9. It's a marker for chondrocytes and pre-chondrocytes. So you can see nothing of these cells are located there. But if we do exactly the same, but just inject amoxicillin or kind of label those cells exactly the same time point, but analyze just few days later, that's what we see. So same green cells we now can see inside a cartilage. So what does it mean? It means that they originated from those cells we labeled a few days ago at E11.5, and those we've seen E12.5. And E stands for embryonic day, mouse embryonic day 11.5 and 12.5. So, and you can see that these cells now, the cells we labeled E11.5 are now generated cells which we see in a cartilage. So these are chondrocytes. They are double positive for SOX9 and they are green, meaning that they've been genetically labeled. Same we can do, and Tujuan stains the nerve, the blue one. So same we can do to check what's going on in the bone, and we can see that osteoprogenitors, which we can detect by Ostrix immunolabeling, they're also green, so meaning again that they are originated from Schwann cell precursors. And you can see that in mandible, in rib, in scapula, and one costa staining just adjusted section just to show you that there is a bone minerals in that region. And here we can also see that if we label, we can see the green cells inside the bone area using another staining for osteocytes, which are most abundant cells in the bone. And you can see that a lot of green cells, which means that they originated from Schwann cell precursors, contribute to the bone tissue in the animals. So that allows us to conclude that Schwann cell precursors contribute to generation of osteo and chondroprogenitors. And that's there are basically two major ways in skeletal or in stem cell biology. Currently, it's a lineage tracing, the most convincing one, and single cell sequencing, which Kai Fred pointed out. So, and another example I want to show you again. So, you're already familiar with this mouse or with a Cre-RT strain, with Cre-RT system, right? We inject amoxicillin, we get our gene activated, and reporter gene usually looks like this. So we have a LOXP site surrounding a stop cassette and somewhat fluorescent protein. But now we make it a little bit more complicated, and we put 4 different fluorescent proteins and 4 different LOXP sites. So what's happening here, when Cre recognize them in a random manner, and in a random manner, it will produce different types of fluorescent proteins. So it depends what LOXP site the Cre will bring to another LOXP site. It will make different types of recombinations, as you can see here, and it will give rise, the cell will produce one or another 4 different colors, or one out of 4 different colors. How that can be used? It can be used if we label a stem cell, let's say stem cell population, in this manner, so our stem cells will be of different colors. And accordingly, when they proliferate and give rise to their progeny, those progeny will have the same color because it's on genetic level, and accordingly they will make clones which we will be able to see these different colors. That been first used by Snippert and Han Clever's laboratory in 2010. Professor Fried showed today this for tooth development, and a Nature paper 2014 for Schwann cell precursor was exactly the same system. And now we have it in Moscow, and we have this reporter strain or we will have it in a couple of months in Almazov Center as well. So what did we use it for? So we wanted to see what the stem cell dynamics is in the growth plate. So we take a tissue-specific reporter or tissue-specific carrier for chondrocytes, cross it with a confetti, inject tamoxifen in embryonic time, and analyze in postnatal life in a neonatal. And you can see nice small clones formed within this growing bone. And that's how it looks on a static picture. So you can see the clones are small, there are quite many, and that's on the histological section, just show you what we are looking at. However, situation changed dramatically when we did exactly the same thing, but in a postnatal life. You can see the clones are changed dramatically. They are spanning throughout the growth plate. So they get, if we go to the next slide, they are really getting really large, reaching up to 50 cells, and they are very stable. And we can see those clones for 6, 7, 10 months after we label the initial cells. So, and that analysis and this observation as a core observation plus few additional analysis allowed us to conclude that during embryonic development, the growth occurs based on chondroprogenitors, which are consumed, make small clones, and then that's contribute to the bone growth. But postnatal growth is based on an entirely different principle. There is a stem cell niche formed, and it stabilizes some chondroprogenitors in the form of stem cells, which can renew themselves and can generate stable large clones. So this was an example how this lineage tracing and clonal lineage tracing can be used to address scientific questions. And with this, I'd like to thank you for your attention, my funding bodies, and my research group. Thank you very much.
Andrei, thank you very much. It was really cool and very informative talk. Unfortunately, we don't have a time for the question, but anyway, so once again, thank you everybody in our session. Теперь можно по-русски, наверное. Всем спасибо.
Now I can move over to Russian, so thank you everyone. Thank you. I think we can wrap up, and we received just plenty of information. Thanks for your participation.